The next step in recombinant DNA technology is insertion of that recombinant DNA into a competent host cell or an organism. So far, we learned about the steps using enzymes by which the DNA is isolated. Then the DNA is fragmented using restriction enzymes, and they are separated by gel electrophoresis. And after that, they are amplified using PCR. Okay, and a recombinant DNA is formed by combining it with a vector using DNA ligase. Then this recombinant DNA has to be inserted into a host cell or an organism. And if a recombinant DNA, we had learned about the requirements of a vector. Remember the selectable markers, which is genes which is coding for antibiotic resistance. So if a recombinant DNA is bearing the ampicillin resistant gene and it is transferred to an E. coli, this recombinant plasmid or the vector is having the gene for ampicillin resistance, and it has been introduced into the host, which is an E. coli. The host cells, now these E. coli cells becomes resistant to ampicillin. Then they can be differentiated from the non-transformants. So once they have received a recombinant DNA, this bacteria becomes the transformant, and they can be differentiated from the non-transformants who lack this ampicillin resistant gene by plating them in an ampicillin medium. So which will allow only the transformants to grow and the non-transformants will die. And obtaining the foreign gene product. The ultimate aim, so far we did all these steps in order to obtain a particular gene product, and this gene product can be a desirable protein. When the gene of interest expresses, we get a particular protein, a desirable protein. And if this protein encoding the foreign gene is expressed in a heterologous host, it is called as a recombinant protein. Okay, so the ultimate aim of recombinant DNA technology is to get a desirable protein, and this protein encoding a foreign gene which has been obtained by the expression of a foreign gene in a host organism, in a heterologous host, is called as a recombinant protein. Cells with these foreign genes can be grown in laboratory. They should be cultured. They can be cultured in the laboratory, and the cultures are used to extract the desired protein and purify it by using separation techniques. The cells can also be multiplied in a continuous culture system. So we have to multiply the cells, maintain the foreign gene in the host cell and also transfer it into its progeny cell. So it has to be multiplied. Only then we get the desirable protein, the recombinant protein in large quantities. So the cells can be multiplied in continuous culture system. Here the used medium is drained out. In the cultures, the used medium which has been utilized by the growing or the multiplying host organism should be removed from one side, while fresh medium is added from the other side. Without giving the inputs, the organism or the particular host cannot multiply. So from one side, the used medium is drained out, and again simultaneously on the other side, new medium is input or it is given. It maintains a cell more physiologically active and so produces large biomass. It is more desired protein. So the more the medium is given, okay, the medium is given, they remain physiologically active and they increase in number, that is increase in biomass, and they give the desired protein in large quantities. Cells with a foreign gene are cultured in laboratories or in a continuous culture system, which helps to the cells to multiply in large biomasses, and thus we obtain the desired protein in large quantities. So we have something called as bioreactors. What are these bioreactors? Bioreactors are large vessels in which the raw materials are biologically converted into specific products. The raw materials are converted into products, enzymes, using microbial plant, animal or human cells. Okay, so some reactants along with the host cell, that can be a microbe, it can be a plant cell, animal cell, or a human cell. They are converted into suitable products, enzymes, okay? And these vessels which helps in this process is called as bioreactors. It helps to obtain the foreign gene product. Bioreactors are used to produce large quantities of products. They can process 100 to 1000 liters of the cultures. A bioreactor, it provides an optimal growth condition for the host cell to multiply, like the temperature, pH, substrate, salts, vitamins, oxygen, all the suitable conditions which is necessary for the organism to multiply. Growth conditions are provided so that we get the desired product. The most commonly used bioreactor is the stirred type, stirred tank reactor. There are two types of stirred tank reactor. They are the simple stirred tank reactor. This is a simple stirred tank reactor, and this is a sparked stirred tank reactor, through which sterile air bubbles are sparked. Okay, so that's a peculiarity of the sparked stirred tank reactor, in which the air bubbles can be sparked or it can be input at a high speed. Usually the bioreactors, they are cylindrical and the base is curved so that it helps in mixing up of all the reactants. And also when it is stirred, it helps in mixing up with oxygen, uniform mixing up with oxygen, and alternatively the air can be bubbled through the reactor. What are the parts of a bioreactor? An agitator system, an oxygen delivery system, a form control system, a temperature control system, pH control system, and sampling ports for periodic withdrawal of the culture. So withdrawal of the culture, we have sample ports through which it can be removed. And the last step involved in recombinant DNA technology is downstreaming process. It is a series of processes such as separation and purification of the product, of the gene product. After the biosynthetic stage, after it has been cultured in large vessels like bioreactors, it has to be separated and purified. And this process is called as downstreaming. The product is formulated with suitable preservatives. Preservatives, we know it increases its longevity. And such formulations undergoes clinical trials. It cannot be used with initially itself. It has to undergo a series of clinical trials and strict quality controlling testing, and then only it can be released as a product. So last step is downstreaming processing. So far we learned about the steps involved in recombinant DNA technology. Thank you.